So in this video we're going to look at magnification, and magnification is basically how much bigger than the specimen it is. So your image, how much bigger that image is compared with the specimen. So your image is whatever you're looking at, so it could be down the microscope, or it could be a printed off picture. So, for example, if something that you measure, if something is, say, 1 centimeter, and you look at it through a magnifying glass or look at it through a microscope with 100 times magnification, it's going to look, or the image that you would see would be 100 centimeters in size. So that's basically what we're trying to work out by looking at magnification. If something is 1 centimeter, you've got 100 times magnification, your image, that will be 100 centimeters, because that will be magnified up. You've made it bigger, 100 times bigger than it actually is. In terms of calculating magnification, it's quite an easy calculation. So if we just write down the equation, basically magnification is equal to what's called the length of the image, so that's what you are looking at, over the length of the specimen. So that's the actual length, that's the actual size of the object or organism or whatever you're looking at, that's its actual size. Now if you can rearrange equations, then that's dead easy. Just magnification, it's the length of the image over the length of the specimen, and quite often in an exam paper they will give you a mark simply for quoting, being able to write down that equation. Now if you struggle with maths, or if you struggle at rearranging equations, then there is a formula pyramid. So that basically starts off with length of the image at the top, and then you've got magnification, and you've got length of the specimen, and that can be put into a neat formula pyramid like that. And then what you do with that, if you wanted to work out, say, the length of the specimen, you'd put your finger over that, usually draw it out, and then you work out that the length of the specimen is equal to the length of the image divided by the magnification. Or if you want to work out the length of the image, then it's magnification times length of specimen. Or if you wanted to work out the magnification, then it's the length of the image divided by the length of the specimen. And that's quite an easy way if you struggle to rearrange formula in an exam and you just want to learn something to be able to do magnification questions. Now if you do use this, then I recommend you write it out properly in this sort of magnification equals length of image over length of specimen, just to make it super clear to the examiner that you know what you're trying to work out on what the two things that you need to measure or get given. Now it's going to be pretty obvious in most exam questions, because you're going to be given something, you'll either have to measure the length of the image, or you'll get given the length of the image, given the length of the specimen, and be asked to calculate the magnification, or get given two of those things and asked to work out the other. And it's a basic question, a basic kind of calculation question found quite common in biology. And once you can do these, and once you know this formula and how to use it, then it's easy marks, and it's very quick marks as well. It's something you're not having to think about, you can just go out and do it. So the first example of these we're going to look at is calculating magnification. So we're going to work through an example. I'm going to give you the question first, so if you want to stop the video at that point and then try and have a go looking at the stuff before, then you can do, but then I'll go through it. So basically the question is, the image, so the magnified image is 10 millimeters wide, and the specimen is 0.1 millimeters wide. Now usually in an exam question you won't get given it this clearly, but We're just testing the basics of being able to work out that equation. So, if you want to do it, then just stop the video now. Okay, so to work this out, you're obviously going to have to work out what the magnification. So, just to recap, magnification is equal to the length of the image divided by the length of the specimen. Now, to do that then, so that is equal to the length of the image. We know that is 10 millimeters. So put 10 in there. These units are the same. They're both millimeters. Okay, so both 10 millimeters for the image and 0.1 millimeters for the specimen. So it's going to be 10 divided by 0.1. Now 10 divided by 0.1 is equal to 100. So we know that it is a 100 times magnification. You'd usually get in an exam, you'd normally have something which came out to quite a nice answer. If you got something like 68.3, then I'd always go back and check it just to make sure that you've got the calculation correct, that you've not divided, that you've not switched the length of the specimen and the length of the image. So we're usually looking for a sensible answer. The second thing we're going to look at is calculating the length. So this is calculating the length of the specimen. Now, we're going to start off with something which the length is going to be equal to length of the image is going to be equal to 5 millimeters, and the magnification is going to be equal to 50. So if you want to do this, then just stop the video now. The basics of this, you're going to have to rearrange that original formula or use that formula pyramid. What you should have worked out, gotten from the formula, is that the length of the specimen is equal to the length of the image divided by the magnification. So those are our two values, and we know both of those. So that is equal to the length of the image, which is 5, divided by the magnification, which is 50, which gives us a length of image. And remember, we're doing this length of image in millimeters, so we're going to carry that units across. That's going to be 0.1 millimeters. Now always remember the units. If you've done a calculation like this, then your length of the image, whatever units that is in, then that will be your answer. If it's asking for a different unit, then you then need to convert this 0.1 into a different unit. So if you need it in centimeters or nanometers or micrometers, then you're going to have to do another calculation to convert that into the right unit. So the last example we're going to do is calculating the length of the image. So to calculate the length of the image, we're obviously going to need the length of the specimen and the magnification. So if we say that we know that the specimen is 0.2 millimeters wide, and we know that the magnification is going to be 100. So again, if you want to try and work out the calculation to work out the length of the image, then just stop the video now and then come back to it. So the first thing, again, you're going to have to identify, rearrange that equation. So what you should get is that the length of the image is equal to the magnification times the length of specimen. So that is your rearranged equation. Now, if we know that the magnification is 100, and then we know that the length of the specimen is 0.2 millimeters, Then we're going to do 100 times 0.2, which is equal to 20 millimeters, because we've used millimeters as our specimen, so millimeters is going to be the unit that we're going to have down there. Again, if you need to do a calculation, then to change your units, to change what it's measured in, then you'll have to do another calculation to change it from this 20 into whichever bit. Thanks for watching that video. If you want more videos, more how-tos, more articles on how to study and how to study science, then go to my website decaffeinatedtalk.com. If you want to get in contact with me, then leave a comment underneath this video or via the form on the website decaffeinatedtalk.com.